to today's EndoCompass webinar. Maybe can you show my slides, maybe?
You want to know? Okay. Sure.
Okay, thank you. The webinar today is entitled Endocrinology and the Environment, the Exposome of Endocrine Disease. I'm Anita Hoppe-Poulega. I'm a professor of pediatric endocrinology in the Erasmus University in Rotterdam, and I will be sharing this webinar today. The next webinar I want to mention is the one next week on Tuesday, November 21st, about the topic, the value of artificial intelligence for endocrinology. That webinar will be chaired by the president of the ESE, Professor Jérôme Bettera. So today we will be updated about the relationship between our environment and the endocrine health, an increasingly vital topic in our field and in the European science landscape in general. May I have this next one, please? The EndoCompass project aims to identify the most crucial priorities in endocrinology and the endocrine science that hold
for nation representation. We've had a new guideline which we're revising soon.
I hear something, but maybe I continue. This is important because it has the potential to dramatically improve the health across Europe. That is, of course, our main aim. This project is gathering many stakeholders to generate a vision and also a plan for endocrine science in the coming years, with the aim to share these with European and national research and health policy makers and also funding bodies. Next one, please. There are also several secondary objectives. We aim to articulate the research priorities towards other funding mechanisms, ensuring that our scientific explorations are well supported and sustainable, to align our endocrine community around a common vision, fostering a united approach to tackle the challenges we face in the endocrine health, and to demonstrate the attractiveness of endocrine research for early career researchers. Highlighting the dynamic and the impactful nature of our work. And then, last, to provide a clear vision to other stakeholders, including the industry, on areas where the healthcare need is high and there are significant opportunities for the development. We will have several deliverables. A detailed roadmap paper in an academic journal, like the European Journal of Endocrinology, open access, also to have research and health policy summaries derived from the roadmap paper, and to have an online communication and dissemination platform for different target groups. The EndoCompass Research Roadmap is a joint project, joint efforts, maybe, yes, okay, of the European Society for Pediatric Endocrinology and European Society of Endocrinology. You see them on the top. And there are also other partners, and you can see these on the right side. They also have, we have collaboration for certain fields with those on the right side. The project itself consists of 9 areas in endocrinology, capturing the diverse and interconnected scientific areas. Next, please. The agenda of today is as follows. First, we will have a presentation by Dr. Julia Mackiewicz, who will present the European Environment and Health Portfolio, Policies and Research.
Right, thank you.
Then the next 2 presenters are George Downward and Sylvain Sebert. They will share with us their great experience in the field of exposome and endocrine and other diseases. Next one, please. Oh, sorry, I have to mention there is also Q&A at the end. So no Q&A after the lectures, but at the end, please. Next one. So George Downward, he's an esteemed assistant professor at the University of Utrecht in the Netherlands. He is an MD, PhD with a background in clinical medicine and global health and epidemiology. And in his today talk, George will provide us with conceptual overview of the exposome and its relation to endocrine science, drawing from his extensive research experience and his work with the EXPANSE project. This insight will be invaluable to our understanding of how environmental factors influence endocrine disorders. Next, please. The next speaker is then Professor Sylvain Sebert, professor at Oulu University in Finland. He has significantly contributed to our understanding of the epidemiology of the course of life. Today, he will be introducing the LongITools and EN-Data catalog. These tools are very important for future research and the development of science-based health policies. Sylvain's expertise will guide us through the potential of these tools for advancing our fields. Then the next one, then I'm privileged to introduce to you the first speaker, Dr. Julia Matskovich, who is an expert in European funding for research and innovation projects, with more than 20 years of experience in international collaborations. She is a biomedical researcher by background and is based in Brussels, and she shares her time between working as a research partnership manager for EU funding for the Brunel University London, with being an independent EU funding advisor. So very welcome, Dr. Matskovich. We are looking forward to your lecture.
Thank you very much for presentation, and thank you very much for opportunity to speak here. And just to add to introduction, kindly presented by Anita, that I was involved in helping with EndoCompass project for some time, but particularly about this environment and health dimension, but also talking about artificial intelligence, which you mentioned as the next webinar. So just next slide, please. So we are all aware of complexity of physical and logistic environment health research and health care provision operates in. Today we are going to hear about environmental determinants of health and disease, but why we are talking about environment and health at all, why that is important. Can I have slide? Yeah, next slide, please. So indeed, the environment is powerful determinant of health and well-being. And if you look at this slide, you can see that World Health Organization estimates that approximately more than 12 million deaths by each year are attributable to environmental risk health. And in Europe, this figure is about 20% of deaths per year. And environmental factors such as air, soil, and water, climate change, exposure to chemicals, they all have significant influence into risk and incidence of disease. So clearly something needs to be done, clearly something needs to be researched. And in my presentation, I'm going to present some of European Commission or European policies and initiatives focused on mitigating this risk or detrimental influence. So can I have please next slide? So well, we all live in era of climate change. So in this context, European Commission was doing some work, and there was several legislation. It's all still in development, but we have European climate law, which makes adaptation to climate change long term for Europe to become a climate resilient society, fully adapted to the impact of climate change. And there are some targets set for reducing net greenhouse emission. But importantly, there is also some kind of measures, sometimes of measures to measure effects of climate change, such as European Climate and Health Observatory, which is already operating. And hopefully next year we get European climate risk assessment. So many policies connected to climate change, they are connected to Green Deal for European economy. And if I can have the next slide, please. So the European Green Deal. European Green Deal is basically the Commission one of flagship initiatives. And it aims to set Europe to pass on green transition, and most of the initiative regulation can be seen as part of Green Deal. Big part of Green Deal is circular economy, which set up production consumption, basically going circular, going back, and saving environment and minimizing losses. But so major goal of Green Deal is towards climate neutrality, but also towards saving and healing planet and ultimately supporting human health and well-being. Next slide, please. Right, so let's have a look what kind of policy we have. There is Zero Pollution Act, which Zero Pollution Action Plan, which aimed at improving air quality, reducing waste plastic litter, improving soil quality, biodiversity, diminishing transport noise, and significantly reducing waste generation. That's kind of overall policies, but if we go into particular pollutants, so to say, can I have next slide, please? There is certain legislation initiative concerning air and water. For example, directive on ambient air quality and clean air for Europe, clean air policy package, drinking water directive. Interestingly and importantly, there is also there is some standards. There are measures, there are air quality standards and watch list for quality of water. Connected to next initiative I'm going to talk about, there is monitoring closely, again, this is kind of enshrined in law, potential presence of two endocrine disruptive compounds, beta-estradiol and non-polyphenol. So next slide, please. Then brings me to something which I believe very close to the heart of ESC society. This is regulation of registration, evaluation, authorization, restriction on chemicals, REACH. This is REACH is about regulation on chemicals, but it's actually about also ensuring a high level of protection of human health and environment against harmful substances. And the European Society of Endocrinology took active part of consultation on the roadmap for chemical strategy for sustainability, but it's also in collaboration with other societies submitted a petition to the European Commission to take urgent act to revision of REACH, because I believe there is more information is available to update that, particularly I believe regarding endocrine disruptors. So next slide, please. Then there is plastic strategy. Plastic strategy, most of this information is available on slide, but it's actually part again of circular economy action plan. And building a lot of measures to reduce plastic waste, like new rules of packaging, separate collection plastic waste, restrictive use of microplastic, bio-based biodegradable plastic, sea plastic. Again, what regulation feeding into it is restriction addressing added microplastic. There is regulation, suggestion of regulation of pellet loss, bio-based biodegradable, compostable plastic, and single-use plastic. So this policies is still in development, but there is a lot of work going in this direction. Next slide, please. Now, due to increasing environmental challenge and need to protect citizen health from the impact of environmental degradation, European Union, sorry, not Commission, has established extensive framework, thematic initiatives and regulatory actions related to environmental and health. European Union also been provided increasing financial support for research and innovation in this area. Why it is important? Because just to come up with any legislation or any law, you need scientific evidence. And that has been produced by European funded project in this area. So there is three major parts of this big package of various projects or pillars. One is European Green Deal. And I'm sorry, I forgot to mention a part of the European Green Deal is also a framework for endocrine disruptors, which is, again, I believe society is very actively participating in. Another one is urban health, and then public and occupational health and safety. So next slide, please. One of things that Commission is doing now very extensively is having a clustering project. So basically the idea is just to bring together portfolio of projects, collaborating together, addressing similar research area, and they are called clusters of project and aims to promote synergies, knowledge sharing, increase societal and policy impact of this research. All right. Okay, and so I'm not going to name all the clusters, just to name European Human Exposome. This is the one where our speakers are going to tell you more about, but you can see it later on yourself. So next one, please. On chemicals, we also have a massive European Human Biomonitoring Initiative, which is now finished, produced a lot of very interesting results. And follow-up is European Partnership for Assessment of Risk of Chemicals, which will support chemical risk assessment and risk management. And finally, if I can have next slide. So, where is the research funding coming from? Just to show you that cluster one, this Horizon Europe, cluster one health have specific destination called living and working health promoting environment. This is just an example of kind of research which is funding there. And next one. And another one is missions in Horizon Europe, which is new things in Horizon Europe funding. So this is massive kind of initiatives. One of them, mission of climate change, which also includes resilience, research and resilience of an impacts of climate change, and then mission of climate neutral and smart cities, which among other things foster measures to improve people's health. So that's probably a very brief snapshot. And just to say then that there will be a lot of opportunities to do research in this area. And I just encourage you to have a look on the ESC website, where is European Research Funding. We are publishing new opportunities and calls on a regular basis. Thank you.
Okay, thank you very much, Julia. The Q&A will be at the end of the session. So maybe we continue then. Is that possible with George?
Yes, absolutely. Let me just quickly share my screen. There we go. I think it looks good. Please shout out if it doesn't.
Yes.
I see nodding. Thank you. Thank you, and thank you very much for inviting me to come speak. So my name is George Downward, and as was said during the introduction, I am based in the Netherlands, where I work at Utrecht University and the University Medical Center Utrecht, which is a large teaching hospital. I do a lot of work in the spheres of exposome research and planetary health. So it's great to have this opportunity to come and talk to you all and give you something of a conceptual overview of the exposome as a whole, try and break it down into some of its components, and then throughout I'm going to be trying to link to applications that already exist in relation to endocrine science. So yeah, as I already said, I'm just going to start with some brief background to the exposome, some definitions, and then talk very briefly about how we measure or quantify the exposome, and then bring all of these things, these backgrounds, these definitions, and these measuring approaches into sort of the exposome profiles. So just to get started, I don't think this is necessarily news to anybody, but overall, heritability alone or genetics alone typically gives something of an incomplete overview of disease, right? The genetic contributors to disease causation obviously varies from disease to disease, but often leaves a lot of questions in the etiological framework or the etiological window. And overall, we tend to estimate that the environment, either alone or in some form of gene-environment interaction, contributes to between 85 to 95% of the overall disease burden. So environmental exposures are therefore incredibly important when we're trying to examine diseases, work out causes, and therefore interventions, protection strategies, and so on. But the environment itself isn't a simple thing. Oh, apologies. It instead represents a wide variety of sources, contributing factors, constituents, pollutants, things that work synergistically or antagonistically, all over the place. So what we end up with is this very complicated combination of features. Sometimes these features have common sources, sometimes they don't. Sometimes they work together, sometimes they're antagonistic with each other. And across all of these environmental sources, we have this wide variety of internal and external effects. So when we really want to try and dig into how these features are contributing to human health, we need some form of systematic way to try and totally examine these features and what they're doing to us. And the exposome concept is one such approach. And this is something that we've been utilizing quite a lot, especially here in Europe. And as was already mentioned in introductions, one of the larger projects that's going on here in the Netherlands is the EXPANSE project, which is a large European-based exposome mapping project to try and develop detailed exposome information on people throughout the continent. Now, the name of the exposome, we typically give credit to Chris Wildworth back in 2005. And from the name exposome, the ome is lifted very quickly from the genome concept. And whereas that was trying to map the entire human genome, the exposome concept is instead trying to map or to model the entirety of human exposures, typically from conception, or as we're learning more and more, from preconception to birth, to adulthood, to old age, and then to death. And the goal, the purpose, is to measure, model all of the exposures someone experiences throughout the life course, and then to try and link those exposures the internal biological pathways, biological mechanisms, ideally even at a pre-disease stage, but otherwise to then follow those pathways through to health phenotypes for disease prediction or targeting areas for intervention. Now, of course, this is a very broad topic then, and one that can sometimes be a bit daunting to approach directly, lots and lots of different pieces. So to break it down to sort of be a bit more digestible, and two, to also be a bit more systematic in how we approach it. We typically try to divide the concept first into the internal exposome, which is where we're referring to those internal biological processes that are occurring inside of us as a result of our external exposures. And then as the opposite to the internal exposome, we talk about the external exposome, all of those features that we encounter in our day-to-day life. We then break the external exposome itself down into 4 micro-environments, or at least this is the approach that we're using here. The social environment, food lifestyle environment, built environment, physicochemical environment, to give us some distinction between exposures, but also to allow us to continue this somewhat systematic approach. I should point out, of course, that even though we break these into groups, they're not completely separate. It's a bit of an artificial division, but it gives us a framework in which we can examine a wide variety of exposures across people. So what I'm going to do for the next few slides is quickly break down each of these environments and then give a couple of examples of how these environments are already being used in various studies of endocrinology. To start with very briefly as the social environment, which is, in reality, it's your very classical social determinants of health that gets used a lot in a lot of epidemiological studies anyway, right? Components such as socioeconomic structure, socioeconomic status, family structure, cultural makeup, social supports, inequities, as well as individual and area level socioeconomic features. And these sort of systems, such as social supports, socioeconomic status, has a wide variety of impacts, of course, for a variety of disease states. And a really nice example came out of Sweden a few years ago, looking at the role of neighborhood deprivation status on diabetes risk. This was sort of a nice little quasi-experiment that was run, looking at refugees that were coming into Sweden and looking at where they were being distributed throughout the country, looking at the neighborhood level of deprivation they had. And there they observed that refugees that were placed in areas of high levels of social deprivation had an increased risk of development of diabetes compared to those who are in a low level of social deprivation. If we move from the social environment to food lifestyle environment, this is another fairly sort of classical lifestyle type of approach, where there we're looking very often at individual activities, diet, exercise, smoking, alcohol, and so forth. But then we're also complementing that with a lot of environmental or surrounding environmental features, such as the proximity and availability of a variety of resources. And this is typically talking about food, but can also be used to talk about things like gymnasiums or sports halls. And this includes a variety of features such as distance, density, affordability of resources, how certain things are promoted, and also whether someone is living in what's referred to as a food desert or a food swamp. A nice example of how these can be used actually came out of the Netherlands a few years ago, looking at the density of fast food outlets within 100 meters, 400 meters, a kilometer and 1.5 kilometers. And it's then relationship with the development or not the development, it's relationship with diabetes prevalence. This was a cross-sectional study. And here we found, or here the group found, that similar to the refugee study from Sweden, that living within a high density of fast food environments had an increased likelihood of also having prevalent diabetes. Now, of course, this sort of finding sort of will vary in its replicability across places. The Netherlands is a very dense place, so people have a lot of food environments very close, and the rise of delivery services sort of muddies the water a little. But this is something of an example of how the food available to people, the food around people can influence their health. If we keep moving, and we go from the more social to something of the more concrete, if you excuse the pun, through the built environment, which largely refers to the sort of the characteristics of the cities and the towns or the communities that we live in. And here we're often looking at features such as the streetscapes, the urban spaces, the presence of green spaces, the parks, blue spaces, so water, and grey spaces, your large urban sites, as well as how accessible, how walkable your cities are, and how certain features are accessible, such as healthcare accessibility. A really useful example I found for endocrinology has to be the light at night feature, part of the built environment. There's a growing evidence base that excessive exposure to light at night is having a strong function, or having a noticeable function as an endocrine disruptor, especially through the dampening of melatonin rhythms, which then will subsequently have sort of disruptive effects on human health. And this can be seen both sort of in a conceptual matter, but also using some biological studies, some biological data, where high exposure to bright light compared to dim light can have a profound difference in salivary melatonin concentrations. So here again, we're seeing components of the physical environment in which we live having clear impacts upon our internal biological processes. As we round out the external environments that people experience, we come to the physicochemical environment, and this is kind of your classical environmental exposures. When people think about environmental epidemiology or environmental health, these are the things that people typically seem to consider. Contaminants in the air, water, soil, and of course, more importantly, given ongoing climate change, changes in temperature and extreme temperature events. This is also where a lot of endocrine disrupting compounds are measured and reported in the literature. Frequently, when people talk about endocrine disruptors, right, people tend to think of sort of plastic compounds, PCBs, flame retardants, and these sorts of things, which sort of have a very clear or very well recognized endocrinological effect. But I wanted also to take advantage of you as an audience to point out that it's not just plastics or not just classical endocrine disrupting compounds that are also relevant for endocrinological effects. Even some of the more classical air pollutants, such as particulate matter and nitrogen dioxide, which are some of the more heavily studied components of air quality, some of the more heavily regulated components of air quality, can have a wide range of endocrinological effects, such as this review, which was studying air pollutants and finding quite consistently that ongoing exposure to contaminated air has a thyrotoxic effect. And then as we go from all of these external processes, all these external exposures, we then come inwards to what the biological responses are. And of course, this is a very, very broad topic, because the number of internal biological processes are virtually countless. And I just wanted to highlight here a way in which we can start to look at this by highlighting some work that a PhD student of mine is doing. This isn't an endocrinological study. This is a lung cancer study. But essentially what my student has done here is he's taken what we call a metabolomic profile, where we've analyzed thousands upon thousands of compounds using high-resolution mass spectrometry, and essentially comparing two exposure groups, high exposed versus low exposed. And for each of those, you're getting a p-value for that exposure contrast. And then because you're comparing thousands upon thousands upon thousands of compounds, you do have to do some correction for false positive rates, because your traditional p is less than 0.05 does not work when you're doing thousands upon thousands of comparisons. So essentially, this is based upon our false discovery expectation. Anything above this line is our new expectation for statistical significance. From this work, you can identify individual compounds, internal compounds that are significantly associated with specific environmental exposures, which can then be linked in their own way to metabolic or internal biological pathways. And in this case, we're trying to link some of these pathways to lung cancer risk, but it is also being applied for a variety of other settings, such as cardiovascular disease and respiratory disease. I'm coming up to the end of things, and thanks everyone for listening. A couple of slides on just how we measure things, because there's a lot of stuff, a lot of information in the exposome. So we have to use a lot of different approaches to try and measure it. One approach we commonly use, and this is something the EXPANSE project itself is incredibly strong in, is the use of geospatial models. So collecting a lot of data that already exists or that has already been collected, or collecting new data and using it to make these models, but to then generate detailed models of a variety of pollutants and environmental features that people can then come back to and reference again and again and again. And this is where a lot of my work is done, is looking at these geospatial models and intersecting them with residential information and using them in studies of health. There's also, of course, a lot of great stuff that gets done with wearable devices, passive samplers, active samplers, smartphone apps. The technology is just in an absolute boom right now. So it provides a huge range of possibilities for personal monitoring of exposures and activities. And then what you do with all of that is you can then build all of that to start to produce these exposome profiles for people. You can combine all of this detailed information on their exposures and their experiences across each of these external microenvironments in combination with the internal metabolomic or other omic type data. They get very, very detailed individual profiles, which you can then use for research, advocacy, interventions, and so forth. This is just one such example of how this information can be put together. This is a public-facing resource through the Dutch Environmental Agency called Check Your Plek, where you can put in your address and you get quite a detailed breakdown of your various exposures. So to summarize what we've just covered, so the exposome itself as a concept aims to quantify the entirety of exposure across the life course. And then we can divide that into different microenvironments to allow a more systematic approach. And then integrating those environments gives us a very high-level overview for individualized profiles, which we can then use for multiple applications, including epidemiological studies, advocacy, risk identification, and so on. And of that, thank you very much for your time. I will stop sharing. If there's questions, we can have questions at the end.
Okay. Thank you very much, George, for a very good overview. It's completely new for most of us, so it's very good to give that. Thank you very much. No, so as you mentioned, no Q&A now, but I think we continue nu with the lecture by Sylvain Sebert. It's coming already. Thank you. Maybe you unmute yourself, Sylvain.
Yes, sorry about that. Thank you very much. you can see my slides correctly.
Yes.
I think that is a yes, and I'll continue. Thank you very much for this opportunity to talk and discuss about the exposome or the role of environment in endocrine disease and endocrine health. I'm very happy to discuss about what we've been doing together with the EXPANSE project and others project as part of the European Human Exposome Network. And here I want to give you a short overview of the projects and the objectives, to give a little bit of flavor of that. And I'd like to share part of the tools that we have been contributing to consolidate, to give you, to give everyone opportunity for maybe more powerful research in the field as identified earlier. So just starting with the LongITools project, which similarly to the project George has been talking about, EXPANSE, LongITools is also a EU project funded in 2020, just at the start of the COVID pandemic, where we've been working on the problem of the exposome. It's a large project, and we received about 12 million euros to work on the problems of cardiovascular and cardiometabolic diseases and the environmental factors underlying it. Those are part of the members of the LongITools consortium. Those are the partners that you can find, and most of them, as you can see, is a group of 15 universities and small and medium enterprise working on the problems, working on developing applications to either study or communicate on the exposome and the exposome approach. In the context of the projects, we started these projects with focusing especially on the problems of cardiovascular and cardiometabolic diseases, as you probably well know them, especially problems of type 2 diabetes, obesity, childhood obesity. Whereas introduced earlier, there is a very, very strong component, a very strong environmental component in the etiology of those diseases that definitely interplay and interact with the genome. And what we really are interested in is that it's the environment plays a role from as early as childhood or fetal development. And if we think about it, most of the environmental factors that seems to be affecting or influencing health seems to be man-made and could be preventable. And we are trying to develop these projects with opportunity to support the environment and health policies as been presented earlier by Julia. So what is our project about? It's a European project where we want to study the interactions between the environment, lifestyle, health determinants in the risk of chronic cardiovascular and metabolic diseases. Especially a lot of work is being done on obesity, on type 2 diabetes, and then on the heart diseases. We've particularly focusing on some area of the exposome, as George has been hinting earlier. It's challenging, and we need to have a systematic approach to try to reduce the environment to some measurable factors, and we are working on the role of air pollution, noise, and built environment. But we are looking at them in a complex systems approach, trying to really model their effects and how they might influence health through different pathways. So, and that's where we got the name from, where especially LongITools is a combination of the problems, where we, especially in this project, we work on longitudinal process, where we are really interested in trying to understand and the changes. We are also trying to work with longitudinal modeling statistics to especially borrow more power from repeated measures and try to move away from cross-sectional observations, so prevalence, and more understanding onset and incidence. We also develop quite a lot of IT-related tools, data servers, fair data and computation and statistics. And then we are trying to develop tools for some applications, some policies and innovations. So in the full title for the project is Dynamic Longitudinal Exposome Trajectories in Cardiovascular and Metabolic Non-Communicable Diseases. So what is the project about? We are really trying to study how exposures to environmental pollutants are linked to an individual birthplace and living location affect the following cardiovascular and metabolic health trajectories. So trajectory is a very important word, and trying to understand especially different types of trajectory, the trajectories related to anthropometry, so body weight, body mass, fat mass, etc. Also glycemic health and the risk of type 2 diabetes, cardiovascular health, and lipid-related health. So through this project, we are trying to combine the problems of the exposome, so trying to understand the exposure as holistically as possible, and also to combine that to a life course approach to understand also whether there are windows of susceptibility, whether there's cumulative effects, and how things develop through the life course and how this contributes to the development of non-communicable diseases. And that's another part that we are doing a lot, is to develop statistical and mathematical models to understand how things work. So as you can see from the chat, there is questions about, yes, there is no associations between food outlet densities and the prevalence of a disease in the neighborhood. But of course, we know that the problems is relatively complex and more complex than that. It's not because you're just exposed to it that you will consume. This is related to social factors. This is related to mental factors. It is related to commercial factors as well that all are influencing people's individual opportunity to live or not healthily. And this is in this project, and I wanted to spend a little bit of time today to explain what we've been doing, especially with the data, because it's a sort of a very prominent field in European research at the moment, where we have been, especially the Horizon 2020, where we've been working since 2014. I think almost at least everything or many of the projects have been working on with health data is about trying to make the data fair and make the data reusable, interoperable, and useful for as many as possible, not just only the researchers who are working on the project. We are trying to build some types of sustainability and legacy with this data. And LongITools EXPANSE EHEN project in general, we have been developing and consolidating data catalog of different types of cohorts, especially birth cohorts, longitudinal studies, register-based cohorts, randomized controlled trials, and biobanks. Of course, this is only a small part of all the landscape of studies that are available in Europe, but we are trying to build it together with more partners, especially as we discussed earlier, this European Human Exposome Network, which at the same time as LongITools, EXPANSE, 9 large projects such as ours have been funded by the European Commissions, which is a very massive amount of euros of taxpayers' money that has been spent on the human exposome, and with some objectives, so that we try to work together as a cluster to get our tools together and to support let's say the greater goods, but especially trying to support this community of research. So those are the 9 projects available and who have been funded all starting in 2020. And I will not go through them, but they are targeting different part of the exposome and different types of diseases and different types of processes. So if you see from the left-hand side, you have the external exposure that are being being studied in the EN clusters. And you can see also on the right-hand side the different types of internal exposures. This is a complex concept for non-epidemiologists, but it has George explained very well. We're talking about the biological response of what is happening in the body, either in response to external exposures, but also as a response of change in our genomes, etc. So there's a lot of work and lots of data trying to handle epigenomic, transcriptomics, proteomics, metabolomics, and other areas of the home. And most of it is indeed following and using the home dimensions to try to have a comprehensive approach and views of the molecular entities or the environmental entities that we are trying to model. And together, it's a huge networks of people who have been working with data, and this is a sort of short snapshot of all the countries that are for the moment covered with the EN network, and that we are trying to work together and to walk through the process of getting these available data and human data fair. So of course, a lot of European countries, almost all the countries have been represented, and I'm not sure why Portugal and Ireland are not agreeing here. And you can see that there's also quite a lot of collaborations with the US, where similar networks of exposome research is being developed. And what we are doing in LongITools, but also what we are trying to do is and this is the same in EXPANSE, athlete projects, and many others. What we have been doing, and this is a work that has been started not just by us, but since 2016, where there's been a large effort of harmonizing data and bringing them into metadata catalog, where we have been collecting and creating data dictionaries and trying to bring those data for future exposome research. But it's not just exposome research, is I'd say any types of research that could make use of it. And of course, and this is a overall process where really it's quite an iterative process where we're trying to build those large data sets and catalogs of data. And as you can see here, we use this facility called Mogenis that has been started with other EU projects where we have been collaborating since 2016. Let me stop sharing for a minute. I'll share my screen with the website showing exactly what the tools look like. So give me a second. I'll now share again. So this is getting you through these European networks, health data and cohort catalogs, where so we have been working together with many to try to map and identify and organize the different data and variables related to exposome. So here you will find many different networks, because also what we are trying to do is not to create a new catalog, but to try to include many different catalogs and map out how the different cohorts or clinical studies have been used. And if I shortly show you how this looks like, you can click, and this is still under development, and you find information about the network itself. And then you can look at what are the different cohorts that have already joined these networks and providing information about their data. So metadata is, of course, we are not yet sharing real data, and we will never. It's mainly about the catalogs of harmonized data. And as you can see here, there's quite a lot of different cohorts and quite substantial sample size from different cohorts across Europe. And when you can continue looking at the different types of variables that have been included and mapped into those catalogs. And I can here you have an expandable list of different things, and you can see that we have been already collecting different types of clinical information. And when things are here, it means that things are being harmonized and that are already available and mapped across many cohorts. You can see that we have been especially working with diabetes, thyroid disorders, and other different types of diseases, and so on. And there is availability of different protocols and so on. I'll stop sharing again and go back to my presentations. Just be a moment to finish, and those just in conclusion, since how you can use this data, or how that can contribute to the research or the plans you have identified already. It's a fair catalog of metadata, so it's data about the data. It includes the included variables have been harmonized mainly across European cohorts, and the protocols for harmonizations are available. And the plan is that actually they are available for new studies to join it, and we have different protocols. It is a federated catalog, so actually we are trying again not to create a new catalog, but to join and to reduce the number of catalogs that are available in the EU data space, and the data can be connected with a federated approach like DataShield, where we can really do some analysis. And this has been supported by European projects and we will continue to do so. Here are the contact of the brains behind those catalogs, and you can also find the websites and the link. This is under preparations, and it's going to be launched later on early next year. And with this, I'm thanking you for your attentions, and sorry if I've been a bit too long.
Thank you.
Okay, thank you very much, Sylvain. I think now we can start. We are exactly on time. We have 20 minutes for Q&A. There are some questions already in the chat, but maybe it would be a good idea when you all open your videos, and when you have a question that you put your hand up. Is that an idea? Then I can see who has a question. In the meantime, I think I can maybe repeat a question that came via the email. It's a general question, and it's up to you, George and Sylvain or Julia, to answer. It says, with regard to the EU climate change goals, as we've heard from Julia, are there ways the exposome is correlated to these goals and measures? Namely, is it possible to track the effect of reaching or not reaching climate change goals to endocrine disease consequences using exposomes, models, and data? A long sentence. So the question is, how is it possible to track the effects of the climate change to the endocrine diseases using exposome models and data?
Well, there are probably two answers to this question. The first thing is probably well, yes, this is the point of the exposome there's a possibility to use different types of climate-related factors to try to understand whether there's a relationship with disease. And definitely the data that have been collected can be of use. The challenge is, one is, how do we conceptualize climate change from exposome data? And that's something that we are working on as part of the climate change cluster, to try to actually understand change and what does it mean? How do we understand climate change? How do we model that? But definitely we can create this type of exposome on climate scores and try to understand this relationship. But it's a long road, definitely a long road, and that's one of the key issue with this exposome is the causality, as people have been questioning, is that here we are doing epidemiology, observing associations. Questions about the mechanisms are absolutely needed, and experimental research or quasi-experimental research are absolutely needed.
May I ask a question myself? I'm a little bit worried that when I have looked at all the papers and the studies on the exposome and what you have presented, it seems, I think it's very obvious that we need very large populations for that in order to unravel the main variables. But many of our disorders, endocrine disorders, are not so, are quite rare. So what do you have any idea or suggestion how we can then still do this type of research? I'm a bit worried about that we will not reach that. For instance, cardiovascular, yes, that is quite frequent, the prevalence of diabetes as well. But what about all these, let's say, bone abnormalities or other things? Shall we really come to the point that we have an idea? Not immediately, but in 30 years? What do you think about that?
I think this is a very good question. And I think this does hit a bit on the head some of the challenges we face with exposome and environmental health research. Because also often, even for cardiovascular disease, as you bring up, the risk effects we're seeing are actually, they're relatively small in an absolute sense. And it's because you've got such a large population that are exposed that you can end up with such a disease burden. So especially now in the EU, where air quality, for example, is getting better, these effects may be getting more subtle. Something that we've been doing more here with the groups I work with has been leveraging a lot of the existing case control studies where a lot of the cases already exist. And then resources like the EXPANSE project, or what Sylvain's described, can then sort of be used, or at least used in an attempt to retrospectively assign exposome exposures and an attempt to try and address some of this challenge you've described. But I agree that it does remain a challenge. But Sylvain, you had something to add as well?
Yes.
Well, yes, it's a challenge, and we have not been able to explain everything in 20 minutes. But what is really important to realize in the exposome, or it's the main shift as well, is to gain precision in the exposome and to try to in understanding environment and health relationship, and is to try to also understand that an environmental factors doesn't act in isolation. So that the exposome models is also trying to incorporate cumulative exposures and how things work in synergy. So how does air pollution or the built environment will contribute to the physical environment, and how people's health will be influenced by multiple different factors. So I think this is also very important, that in the same way that you realize health and environment policy are trying to connect things together, we also have to connect many environmental factors and to have opportunity for integrations. And that's one of the big aspects of exposome research, I think.
Okay, thank you. Anybody else for a question?
Can I ask a question? Yeah, I'm just trying the two questions. One I put in the chat.
Oh, sorry, I didn't see that one.
When Sylvain was talking about all this fantastic data available, I just wonder, is there would be possible to have this data in forthcoming European health data space? Because this is just, yeah, that's probably where they want to keep, well, that is still forthcoming, but they want to keep all the data so everybody can use it in theory. That's a question.
Well, yes, we know that, well, you know about these challenges that are, we would love to, I think everybody would love to. There are still many ethical and legal barriers to be addressed, and I suppose we will see how things develop. At least the information about the data will be more and more available and easy to trace, and whether or not we will have a European data space, I cannot.
Yeah, that's a question mark.
I hope so. I hope so. You know, we have fantastic data indeed here. We hope that we could get them available. There are many challenges. You mean already, if you think about the language, we have the data in described in Finnish here. There's still a lot of work to be done, but it might be available sometimes.
Oh, let's hope.
Yeah. But it's at least now we have. I think we had during the last 10 years realized that moving from my data to our data, and that's really a big leap. I mean, there's no more this PI who controls the access to the data. The data is more easily accessible. It's not a perfect world, but it's better.
May I ask you, do you have an advice to ESE and ESPE for their members? What could be the best next step? Is there an idea to, let's say, to have an initiative to first of all to increase awareness for our I think the members, but maybe next that we try to collect data, maybe not via our society, but to organize that it is directly done to certain collaborative partners, groups. This is a suggestion. Please give your advice. I think that's also why we have this webinar. What would be the best next step for us?
Oh, well, the advice would be to yes, embrace the process of fair data management. and if it's not done yet, they should do it. And to already each researcher should really be mindful of the importance of the data management plan and how much you are seeing all the data that you are collected can be potentially useful as to others. And I think so that they definitely should not try to do a new one if needed, but to try to reach out to those who already started, and yeah, and really to realize that it's the importance, as you know, to when we start moving to environmental factors in relation to diseases, the importance of replication and harmonization and comparison is extremely important, because the endocrine disruptors, they are small effects, so that can be highly biased by different factors.
Okay, thank you. George, do you want to add, or Julia? Maybe you have other advice or confirm? Or...
Not sure how much to add. I think Sylvain's done a very nice overview. I know the EXPANSE project, just regarding data, the EXPANSE project likewise has a strong sort of fair data commitment. But practically a lot of the infrastructure to make data easily available is still needs development. So a lot of data, I mean, it's available, but it's available on request and following review.
I must I only can add, from point of view again of European project, fair data is really big requirement. I'm talking about now next webinar when we're talking about artificial intelligence, again, that needs fair data. So maybe we can have further discussion about it at some point. But I think I saw one hand, but then it disappeared.
No,
maybe.
I didn't see a hand. Is that, sorry, then I've missed it. I didn't see a hand as far as I could see.
There was somebody's, but I think it disappeared now.
Oh, thank you for checking, but I don't see it.
Okay.
Is there anybody who wants to pose a question? Please, this is your chance. No? I think it's quite overwhelming, this, at least for me. I have to take this personally, of course. What is going on, and it's such a different environment than where we work as pediatric and adult endocrinologists. I think, but it's so valuable to hear about it. Thank you very much. I think that nobody has other questions anymore. It's maybe time to close the webinar. Yes, this is your last chance. Thank you very much. It was brilliant, really. Thank you. And then I'm going to close, and have a nice evening. Okay, bye-bye.
Thank you very much for having me.
Thank you.
Thanks,
Have a great night.
Bye.
Bye-bye.